So five tricky questions. So let's see what are the different or the five tricky questions which are usually asked in the competitive exam. So first question is absorb monosaccharide in the intestinal epithelium cell. They exit via which one of the following transporter? The GLUT2, the option basically are the GLUT2, GLUT3, GLUT4 and the GLUT5. So we know that the D glucose and the D galactose are basically transported across the brush border membrane of the small intestine, that is it enterocyte via the sodium D-glucose obviously transporter, and the process of absorbing the monosaccharides such as the glucose and the galactose in the intestinal epithelium cell, it involves the transporter which is known as the GLUT. Okay, so the GLUT is a transporter which is mainly used for the absorption of all these monosaccharide, the glucose and the galactose. Now, among these, the GLUT2 play a key role in the absorption of the monosaccharide from the intestinal lumen into the intestinal epithelial cell. So, from the lumen to the epithelial cell, the GLUT2 is the main transporter. So, the answer will be the GLUT2. The explanation for the this GLUT2 is that they help in the absorption of the intestinal epithelium um, cell. When you consume the carbohydrate in your diet, they are first broken down into the monosaccharide, that is the glucose during the digestion, and these monosaccharide they need to be absorbed from the intestinal lumen, and they are need to be absorbed from the lumen to the epithelial cell so that they can enter inside into the blood stream. Okay, and the GLUT2 transporter is a glucose transporter only, which is embedded in the cell membrane of the intestinal epithelial cell, and it is very unique in that it has the high capacity of transporting the glucose and the other monosaccharide. The transportation of the monosaccharide also take place with this GLUT2 transporter, and also the GLUT2 are basically present at the basolateral membrane. Okay, basolateral membrane. So it facilitates the exit of the monosaccharide, allowing them to move from the epithelial cell into the blood stream. They have a role in the blood sugar regulation, means GLUT2 is not found in the intestine but in the other tissue including the liver. It is not only found in the intestine but also in some other tissues also, such as the liver, the pancreas. In the liver, the GLUT2 is basically involved in the glucose uptake, and in the pancreas glucose uptake, and they really they playing a role of the blood sugar regulation. So in summary we can say that the GLUT2 is a crucial for the transport of the absorbed monosaccharide from the intestinal epithelial cell into the blood stream, which are contributing to the overall regulation of the blood sugar level. So answer is the GLUT2. The second question is the individual chromosomes are seen in which phase of the cell cycle? G0, G1, S and the M phase. So we know that M phase, that is the mitosis of the cell cycle, is specifically the phase in which the chromosomes are seen. Why? Because the M phase is the specifically phase of the cell division where the cell undergoes the mitosis leading to the separation of the chromosomes into the two daughter cell. Okay. And during the mitosis or the M phase, the chromosome usually condense. They usually condense and they become individually visible as the cell prepares for the cell division. So when they condense, they take the formation of the chromosomes only. That is why they are visible in the M phase. While in contrast to the other phase, that is the G naught and the G1 and the S phase, the chromosomes are not as condensed or visible because they are in the state of the replication, in the growth, or in the G naught is the resting stage. So that is why the chromosome is not visible in these stage. Question: the table below, table in the below list the cleavage pattern and the name of species. You have to match the cleavage pattern with that of the species. So the cleavage pattern, we know that it is a series of the mitotic division whereby the enormous volume of the egg cytoplasm is divided into the numerous small nucleated cells, and they are the different pattern of the cleavage pattern. So let's see what pattern are actually present in the species. So isoleucithal bilateral cleavage pattern. In the isoleucithal bilateral pattern, the sparsely distributed yolk sac. Okay, sparsely distributed yolk sac, and these isoleucithal typically is found in the tunicate. So let's see where this isoleucithal bilateral isoleucithal can be seen in most of the species, but the bilateral type of isoleucithal pattern is usually observed in the tunicate. So let's see where is the tunicate. So one A matches with the third, and what are the options given where the A matches with the third? This one. So let's see all other cleavage pattern, it matches with the species which is given in this option, that is the fourth option, is correct or not. So A matches with the tunicate. The second is the mesoleucithal radial. The mesoleucithal radial, they are commonly found in the amphibians such as the frog, and where the yolk is concentrated at one pole of the egg, and the cleavage is the radial type. So mesoleucithal is usually found in the amphibian. So the B goes with the first, and this B is again going with the first. The third one is the centroleucithal, as the name suggests that they are have the central location. The yolk is generally concentrated in the central region, and the cleavage pattern is generally superficial. That is, they are occurring in the periphery region. And these type of cleavage pattern, they are generally seen in the insect. So the C will go with the fourth. Again, the C matches with the fourth. The fourth one is the teloleucithal discoidal. It is a characteristic of the bird and the reptile, and the yolk is concentrated at the one pole only. The telo, as the name suggests, telo means at one end. So at one end, it is concentrated, and basically it is at the vegetative or vegetal pole, and the cleavage occur in the disc-shaped area or the animal pole. So these teloleucithal discoidal, it is observed in the bird. So the D option will go with the second one. So the correct option is fourth, that is A with the third, B with the first, C with the fourth, and D with the second. So this one is the correct option. Next question is, a few organelles that are present in the eukaryotic cell are mentioned below: the centrosome, peroxisome, nucleolus, endosome. Now, which one of the following option they represent the organelle that are not membrane bound? So we know that the uh, endosomes are obviously the membrane bound. The peroxisomes, they are also a membrane bound, but the centrosome and the nucleolus, they are not membrane bound. Okay, so these are the of two organelles which are present in the eukaryotic cell, and they are not the membrane bound. So let's see where is the option is given, the A and the C, the A and the C. So the third option is correct. So we know that the nucleolus is basically involved in the synthesis of the ribosomal rRNA, and it is not surrounded by any membrane. And the centrosome basically they play a role in organizing of the microtubule during the cell division, and it is also a non-membranous. Okay, while on the other hand, the peroxisomes and the endosomes they are the membrane-bound organelles. The peroxisomes they are involved in various metabolic activities such as the lipid metabolism, and endosomes they are responsible for sorting and the transporting of the cellular cargo. Next question is the column A lists the enzyme which is involved in the cell cycle, and in the column B is typically a function is present in the particular cell cycle. So we have to match that which particular enzyme goes with the cell cycle function, and the option basically are given here. So the aura B, we'll start with the last one, the aurora B. The aurora B basically they are involved in the mitotic entry and mitotic entry and they are involved in the mitotic spindle formation as well as in the cytokinesis. So aurora B, the mitotic entry is there and the cytokinesis is there. Two options are there. So let's move to the another enzyme, that is a CDK2. The CDK2 is a cycling dependent kinase 1, and the cell cycle stage where actually it work is the mitotic entry. Again, the two option clashes. So it triggers the entry into the mitosis, that is the M phase, and also regulate the various processes during the mitosis. But this the decay 2 basically it goes with the mitotic entry because aura B is can be present in the cytokinesis also. Okay. Next is the APC and the CDC20, that is the anaphase promoting complex or the cyclosome with the CDC20. So as the name suggests that anaphase promoting complex, so obviously they are present in the anaphase is onset only. So this will go with this. The anaphase onset because it regulates the transition from the metaphase to the anaphase by targeting the protein for the degradation. The last one is the CDK1. Um, sorry, there is a mistake. The CDK1 actually goes with the mitotic entry. Okay, this. Yeah, the CDK1 will go with the mitotic entry. Okay, because it triggers the entry into the mitosis, the M phase, and regulate the various processes during the mitosis. While the CDK2, it is actually working in the S phase entry because it regulates the progression from the G1 to the S phase. So the A matches with the third, where the A matches this option showing the A matches with the third and the B matches with the fourth, the C matches with the second and the D matches with the first. So option second is correct.